Hi, I'm Rajnish.
And I'm Bridget,
Welcome to Science.
the podcast where reality matters.
Today we have a really wonderful guest. He is the professor of neurophysiology at School of Medicine at Stanford University. Bruce MacIver, Dr. Bruce MacIver, it's so nice to have you on this podcast, and I'm excited about talking about anesthesia and the work that you're doing, and also about consciousness and the recent meeting that we attended together. But before we get started with all of that, we'd love to know something about yourself.
Well, I've been at Stanford about 30 years, and it's the first job I had after graduating with my PhD. I came down here as a postdoc and worked for 2 years in an anesthesia lab and then 2 years in a pain lab. And then I started my own lab, a neuropharmacology lab, immediately after that. And my work's focused on understanding brain mechanisms underlying consciousness, somewhat interested in pain perception and perception in general. But mostly the last few years, it's been about how people lose and regain consciousness, for example, at night when we fall asleep and wake up in the morning. And I've been using various anesthetics and sedatives to induce loss of consciousness, and all the time recording brain signals, either from inside of rodent brains or at the surface of people's brains. And more recently, we're even recording from patients that have electrodes implanted in their brains to monitor changes in neural activity across the divide of consciousness and unconsciousness, and then, of course, upon recovery. Um, so that's basically a short bio. I was uh, uh, raised in Canada and the United States and went to high school in Dallas and back to Canada for university. And then, like I said, after graduating with a PhD, I came directly to Stanford and have really enjoyed living in California. It's a great place to be. Um, so, shall we talk about the meeting?
Absolutely.
Yeah.
Yeah.
Yep. So, Rajnish and I first were introduced to each other by a guy who runs a consciousness meeting, has done for 30 years almost, 25 years at least. And every other year it's in Tucson, and in alternate years it's somewhere in Europe. Last year it was in Interlaken, and I think next year it's going to be in Sicily. So you get a interesting group of people at consciousness meetings. They range from neuroscientists like myself to uh, people who are clinically interested, like anesthesiologists, obviously, and psychiatrists, neurologists. And then you get the full spectrum of people um, that come from parapsychology and from various uh, philosophical disciplines, for example. So, very eclectic mix of people. And Rajneesh and I were introduced by the founder of that, Stuart Hameroff, um, because Rajneesh was interested in consciousness in plants. And we got to talking about how we could determine if plants were conscious. And one way would be if you can stop some vital process that plants do with an anesthetic and then show that it recovers after the anesthetic, then that, at least is a correlate of what happens in you and I when we're exposed to anesthetics. We stop thinking and feeling and reacting, but our hearts and livers and kidneys and other important brain functions that control all of that remain intact. And so that's a short how we got introduced. And then this year, we were excited to host a panel of, was it five or six?
I think yes, five. Uh,
Yeah, five of us that were all interested in one aspect or another of consciousness in plants and how plants can influence our consciousness. So, with that, I'll let Rajnish tell us a little bit about the panelists and and what they were thinking.
Yeah, no. So it was a really a good workshop with a focus on plants and consciousness. Bruce led it, and we had Deepak Chopra, who talked about general consciousness. We had Dennis McKenna, who was talking about psychedelics and how plants make chemicals that can alter our consciousness. And then we had Patricia, who was talking about, she's a professor at Arizona University, I think, or Arizona State. She studies native knowledge and native medicine. So she added a really wonderful perspective of how cultural interrelationship with plants and plant produced chemicals helped evolve human thinking and develop cultural aspects. So I thought it was a really good workshop, and we got really good response.
And don't forget, Rajneesh, your own um, presentation that was preliminary results from our research collaboration showing that you can, in fact, anesthetize plants.
Yes.
Right.
So we had designed this experiment in a way that had not been done before, which is to test whether we can use anesthesia to block plants' ability to respond to environmental cues or signals, and whether that block can be transient and then it can be removed as anesthesia wears off. And not only we found that is the case, but with Dr. MacIver's help, we were able to also show that the volume person by person percent, it's similar to the amounts that are used to lose consciousness in humans and also
Oh, wow.
surgical consciousness. So we had very similar range that worked in plants that is used in surgery.
Yeah. And I did see your slides for that as well. And it's actually really incredible to see, the drooping of the leaves, right? That was part of the anesthesia, and then they do recover.
Yeah, it was
Yeah.
phototropism, so it was bending of
Yeah.
seed.
Yeah.
Yeah, yeah, no, it was quite interesting. So
Yeah.
all of this is part of trying to understand what is consciousness. And if I may share something on the screen, and I'll describe it for our listeners who can't see the screen, I would like to just go through this plot that I have seen on Dr. Bruce MacIver's website. So anyone can go to stanford.edu and look for MacIver Lab research, and you will see this plot. So what's interesting to me about this is on one side, on the y-axis, is going from 0 to 10, and it's showing a degree of awareness of environment. So how equipped you are, or how well you are in being aware of the environment. On the x-axis, it's showing level of arousal, so how awake you are in a way. So under anesthesia, obviously you're not that aware. Like if you're in a coma, you're not aware of your environment, and you're not awake. So that's at the very low 0 0 end. And then as we go forward, there is alertness and awakeness. But what interested me in this plot was that even beyond awakeness and alertness, is the mind expansion and LSD experience, which is beyond the awareness of the environment, highest awareness of the environment, and the highest awakeness. So I would like you to tell us a little bit about this, how this may be possible.
I'll point out that on the y-axis, it's the awareness of the environment, but even beyond that, as you get up to the eights and nines and tens, it's self-awareness. And so out on the far end of the far right-hand side of the graph, you're up at a very high level of self-awareness and of alertness, of arousal. So your brain is very stimulated, very attentive, very active. With LSD, you're often hallucinating and going off on little journeys of self-exploration and connectedness with. A lot of people call it a oneness with the universe or a connection to a more cosmic type consciousness. So this plot was adapted from Stephen Lowry's work. He's mostly interested in brain pathology like coma
Mhm.
and these very interesting psychiatric conditions like locked-in syndrome and um, he's treating these patients that can't respond to normal verbal commands or cues and often are only minimally responsive to pinpricks and strong stimuli like that. So he didn't have anything beyond the hyper alert in the awake state. He ended his plot there. And I added the mind expansion in LSD mainly from going to this Tucson Consciousness Conference and hearing stories of the um, psychedelic experiences, not just LSD, but psilocybin and mescaline and um, ayahuasca, all kinds of DMT like drugs. And one thing they all have in common is this feeling of mind expansion and oneness with the universe and intense self awareness and dream states and hallucinations. And um, those go well beyond what we normally experience in our awake uh, mind. Unless you're a little psychotic, then I guess you can hear voices and hallucinate as well. But so anyhow, this is modified from Dr. Lowry's work, and the only additions I made were the awake anesthesia condition because that's something that's of clinical concern to anesthesiologists and surgeons. Um, that he hadn't taken into consideration, but it's a lot like what he calls an inverse zombie um, condition, where you're aware and feeling and conscious, but can't move or report any of your experiences.
So you know
So,
that you're aware of the environment, but you're not awake. That's where that right. Right.
right,
Yeah.
yeah, and that's on the far left and higher up the axes, and and he also didn't have anesthesia extending nearly as far as it does into the awake condition because he's not aware that the low concentrations of these drugs first affect your memory, your ability to learn and remember, before they even start to make you too sleepy,
Mhm.
and like long before you lose consciousness. And there's a whole gradation of drowsy and sleepy, and then you lose consciousness, but can be readily aroused by just your name,
Mhm.
uh, and then you get less and less conscious, where you no longer respond to standardized stimuli like loudly
Right.
saying your name. Can you hear me now, Rajnish? Here, squeeze my finger if you can hear me.
Which is so. This brings me to a important question. First of all, I think even today we don't really have a very good understanding of how actually anesthesia works at the molecular level. We have some good ideas, but we don't really understand how anesthesia removes, let's say, consciousness. But then, as I've learned from you, you've described the state of unconsciousness is very different from sleep. So would you say that we are now talking about three states? One is the sleep state, which you can come out of easily. Then there's the unconscious state, and then what you were describing as the heightened awareness state. Would you say those are three different states?
Oh yes, they differ fundamentally at a neurophysiological level in ways we don't fully understand yet. But clearly, they're involving different brain states that have attributes that are easily segregated. Like you said, in sleep, you're uh, relatively easy to arouse. But when you're in deep sleep, it usually takes more than just calling your name. It might take a little shake on your shoulder or a good like that,
Water.
Uh, yeah. Like,
Yeah.
for example, alcohol intoxication. I'm sure we've all encountered people who were very difficult to arouse
Mhm.
um, after drinking too much, usually martinis. Uh, Anesthesia, of course, goes beyond just loss of consciousness. We want people where they no longer move in response to noxious stimuli, pain. Like
Hmm.
surgical incision is a very nasty stimulus because you're severing pain fibers in the skin
Right.
and, uh, and often stretching tissues underneath that have pain fibers
Yeah.
as well that are what act up when you get distension of your stomach or in cancer patients it's usually stretching of the tissues that causes the cancer pain.
Right.
So so anesthesia starts at the awake state where you're mildly sedated, then you lose your ability to remember, to store information, and to retrieve it, although those are two separable um, uh, processes. So, even just distinguishing between your inability to learn and your ability to remember involve different brain systems, and different drugs change them in different ways.
Hmm.
So, and then you go into more and more sedated until you're at loss of consciousness. But even then, it's what's your endpoint? How do you test consciousness? Is it a gentle thing like a verbal command, or a nasty thing like a smack on the face? So, and then ultimately beyond that is surgical anesthesia, where it has to be very painful to arouse you.
Right. Right.
But getting back to Rajnish's earlier points, we really don't fundamentally know enough about what anesthetics do to the brain to be able to describe how they work.
Right.
Right.
Uh, we know a couple of important things, though. Different classes, chemical classes of anesthetics, work through different mechanisms. They work through different spectra of receptors for neurotransmitters, for example, but also enzymes and ion channels and all kinds of proteins in the brain. And that's what sets anesthetics apart from most other drugs. Typically, a good drug would target a single receptor, like a dopamine receptor for treating Parkinson's disease, for example. And it wouldn't talk to noradrenaline receptors or to ion channels or to any other protein, just that one specific uh, protein in the brain. Anesthetics all talk to more than one protein, and there's often overlap. A lot of them talk to GABA receptors, for example, but not all. And a lot appear to affect excitatory glutamate mediated synaptic transmission by depressing it. But again, not all anesthetics do that. But most anesthetics do a little bit of several things. They'll affect potassium channels, for example, as well as GABA receptors, as well as glutamate synapses. And at glutamate synapses, they can target any of a number of proteins at the actual synapse. For example, they can affect the generation of energy by mitochondria. They target cytochrome 1 in the mitochondrial protein complex to inhibit it and thus reduce ATP and energy levels needed to sustain excitatory connections between neurons. Then, at the actual connections on the presynaptic side, they can block the release of neurotransmitter, of glutamate, by acting on snare proteins, by acting on GABA receptors and ion channels in the presynaptic nerve ending, including potassium channels, sodium channels, calcium channels, all of which are involved in the release of glutamate. And then postsynaptically, some glutamate receptors appear to be quite sensitive and get depressed by anesthetics. And the NMDA and methyl deaspartate type of glutamate receptor, it's a metabotropic, so a slower acting but very important excitatory transmitter system at glutamate synapses. The NMDA receptors appear to be very sensitive to some anesthetics. So you have to think of anesthetics as being dirty drugs. They target multiple proteins, and that's their biggest problem is they produce multiple side effects that are unwanted, like depression of the centers in your brain that control your heart rhythms and can lead to arrhythmias and to um, a loss of control over the function of the heart. They can lower your blood pressure. They can cause you to have all sorts of issues with glandular secretions. They target your kidney, your liver, all this outside of the brain, right? So they're bad drugs. Even our very best drug right now, which is propofol, that has the kind of least amount of nasty side effects, is nasty enough to kill Michael Jackson.
Yeah.
You know, it's a fine line between getting the right amount of propofol to lose
Right.
consciousness and to start shutting down your respiration and causing hemodynamic instability. And that can be enough to kill you, which is why we need anesthesiologists in the operating theater. It's
Mm-hmm.
not enough that the surgeon alone could keep an eye on the patient. Somebody has to be dedicated to keeping an eye on blood pressure, heart rate, respiration. Often we mechanically ventilate patients, so respiration's not an issue.
Right.
But without that, it would be a big issue. Uh,
And
Yeah.
so
some of those receptors like GABA and glutamate, they're homologs of all of those in plants as well. So
right.
it's interesting to and it's easier to navigate the mechanisms in plants than it is in humans and animals. So that's our interest together. Sorry, Bridget, you were going to ask something.
I was just gonna kind of ask about when, for patients who might have anesthesia for many surgeries over a long period of time, is that just like increasing their risk of more and more side effects? I feel like that's something I've heard, but I've never known if that's true. I know that I've had two experiences where I've had to go under anesthesia in like a rush scenario that was very scary, but I don't like, thankfully I didn't have any direct like side effects from it, but it worries me that maybe in the future if I have more scenarios that I have to be under anesthesia, am I more at risk then of, ill side effects in that case?
Yeah. The more anesthesia you get exposed to, the more likely you'll experience bad outcomes, unwanted.
Right.
A common one, especially among older people, is postoperative dementia, where you're
Mm,
you may be spaced out for a week or two after the
wow.
surgery. And unfortunately, old people tend to need more surgeries than youngsters. So Bridget doesn't have to worry too much about it for a while. But Rajnish and
Thankfully.
I are getting close to where that could hit home.
Yeah.
Mm.
On the other side of the spectrum are infants. A lot
Mm,
of kids are born with like congenital heart defects
right.
that you have to do surgery on them or they won't survive. And they may have to undergo multiple surgeries in a fairly short period of time, like months or a couple of years. And in animal studies, there's clear evidence that anesthetics cause harm to the developing brain.
Mm.
They in species from rodents to monkeys, there's clear evidence of apoptotic responses following anesthesia in the young brain. And that means you'll be killing neurons off.
Mm,
Thankfully, because all of our kids are raised in enriched environments where they've got toys and tons of excitement and stimulation, by the time they get to school age, a lot of this, damage um, has worn off. So when you test kids who've had a lot of surgeries very young for their IQ or their scholastic ability or any intellectual type measures you want to use, um, you see subtle effects. So it's not a very worrisome. I mean, we'd like to not harm the brains at all. And there's light at the end of that tunnel. There's hope because there's some new anesthetics that are being developed that don't produce that response in young brains. So another 5, 10 years, we may treat infants a whole lot differently than we do now.
wow.
So and spare them the effects on their brains that we know take place because you can see it in every species,
Right.
but you can't open up kids' brains and look. That's kind of unethical still.
Yeah.
Still, that it's good to hear that there are new anesthetics coming, which may
Yeah.
be more effective and less harmful. Um,
Mhm.
so, I was thinking, since we are talking about anesthesia, one of the things that it is used for is to um, remove consciousness. So, this is how we also first met, to think about what is consciousness. And
Mhm.
obviously, you and I have discussed, there are so many theories of consciousness out there. So I was wondering if you would like to tell us a little bit about what you think of consciousness and the theories that are out there.
Well, Stuart Hameroff, who introduced us, has his own theory of consciousness. That's the only theory that's based in a physical um, world, in physics, if you like, because it deals with quantum um, effects that occur all around us and in our bodies at every moment, because we're made up of stuff that's ultimately behaving in a quantum mechanical system. But anyhow, their theory, this is Stuart Hameroff and Roger Penrose, who won the Nobel Prize last year for his work on black holes. But for a long time now, he's been interested in consciousness and has written books on consciousness. I think Stuart has written numerous papers that include Roger.
Yes.
So it's easy to find their stuff on Google. But their theory is nice because it says consciousness arises out of a physical process where whenever a particle collapses, and they collapse whenever you measure, or it interacts with another particle that determines, for example, whether it had an upspin or a downspin. Whenever that happens, a quantum of consciousness is generated. And they've gone beyond just that causation to how that can be channeled upwards into cells like neurons and networks and brains, because they believe the microtubules, which are the cytoskeletal elements that are in all our cells and form these incredible shapes of neurons with their dendrites and axons, and just an artist couldn't have conceived of more beautiful structures. But all of that structure is determined by the arrangement of microtubules into branches and cell body, etc. So it's quite plausible that they could serve as amplifiers for these quantum um, um, processes that develop consciousness. So little tiny pieces of consciousness are being generated all along microtubules at any given moment. And their idea is that they get stored and transferred. The energy of the conscious quantum is stored and transferred along the microtubules. That would, answer a fundamental question of where does consciousness come from?
Right.
Well, it comes from physical processes like everything else in the universe.
Mhm.
So, and it lends itself to ideas of cosmic consciousness, because it's not just our microtubules that are um, collapsing and are often entangled with other particles throughout the universe, often at light years distance, that can instantaneously um, collapse into a quantum of consciousness. So it's an intriguing theory from that point of view. And Stu knew that Rajnish had uh, assays, measures, ways to measure microtubule function in plants, and actually visualize the microtubules while the processes are taking place. One in particular is phototropism, where if you shine a light, the plant bends towards the light. And that's known to involve the disassembly of microtubules and then the reassembly of them in the at a 90-degree angle. And so we thought, well, let's look at that process of phototropism and see what happens with anesthetics. And something Rajnish didn't mention about our study that sets it apart from all the other plant studies, because believe it or not, there's been a lot of
Yes.
anesthetics applied to
Since
plants.
19th
Wow.
century.
Wow.
Yes, since the late 1800s.
Right.
So anyhow, none of them controlled the concentrations. So
Mm.
the earliest studies, they just poured ether,
Right.
you know, uh, into a sealed container with the plant in it, and achieved very high concentrations of ether. And they may have been irrelevant to what ether does in our brains because it's there at 100 times the concentration. But anyhow, we measured our concentrations. We held them to the physiological range that occurs in our brains when we're anesthetized. And we're surprised to see that the effect on phototropism occurred over that same concentration range from loss of consciousness to anesthesia.
This is what I find exciting about it, because as you described, many of the components at the molecular level, if they are common, then one would expect that the range to block them would be similar. So,
Sure, the binding to the proteins should be nearly identical because they're very similar proteins. So GABA receptors in plants are nearly identical to GABA receptors in us.
That's
And
interesting. Yeah.
so, yeah, you would expect that the binding would be similar, and hence the concentrations needed to affect their function would be similar.
so one thing that after the direction that we are heading in, and I agree with you. I think also the how I think about consciousness and its source and origin. I think the orchestrated objective reduction, the OR, the Stuart Hameroff and Roger Penrose theory makes more sense to me as well. And microtubules are at the heart of that in terms of amplifying consciousness. So one thing that then comes to mind: are there any neurodegenerative diseases or where it is known that microtubules have been impacted? And so is there that direct link of microtubules with loss of consciousness experience? Uh,
Yeah, I don't think there are, Rajnish.
okay.
I'm certainly not aware of any. You know, you think about microtubules, they're so important to life,
Right.
to from dividing cells as we grow
Right.
and build our brains to holding the whole thing together. Uh, it's unlikely that there'd be any room for mutations
Right.
that could lead to
It'll
disease.
be lethal.
Uh, yeah, they're very well conserved proteins, and yeah, I'm certainly not aware of a microtubule associated disease. Like we know, for example, defects in myelination
Uh-huh.
can underlie MS.
Right. Right.
And, you know, obviously there's a strong genetic component to Parkinson's disease, excuse me, and Alzheimer's disease, but they all target other protein systems. I've never heard of a problem with microtubules.
It's an interesting area, and there is a lot to learn and lot to do. So
Much to do.
yes, but it's also very exciting because it's something that we don't understand, and maybe we will never completely understand it, especially.
But one thing we will understand is what anesthetics do to
Yeah,
plants.
yes.
We've got a new project. You tell them about the stromal
Sure. Yes.
thing.
So, as we were just talking about the phototropism, it's a little bit harder to study many of the mechanisms because it's a very sensitive response. But as it happens, plants have many tricks up their sleeves or up their leaves. So one of the tricks that the plants have is that they open and close these stomata, which are these pores that are on the leaves. And as it happens, the stomata also use microtubules to open and close. Microtubules arrange and rearrange in different forms as the stomata open and close. And it's a very important aspect for plants because when the stomata or these pores are open, they allow carbon dioxide to go in and water to go out. So transpiration that pulls all the nutrients from the soils and carbon dioxide that is important for photosynthesis. So it's a very important part of opening and closing these pores. And so we have already have these plants where we can visualize microtubules, and we have a confocal microscope where we are collaborating with a lab, and we'll be able to see three-dimensionally in the leaves what's going on to the
Wow.
microtubules with and without anesthesia as these pores open, or whether they will not open under anesthesia, and how that
Wow.
effect will be. And the best part about plants is that we can order up mutants. So we can order up some genes that are not functioning. So that will allow us then further to investigate which parts, which pathways might be involved in this response.
Mhm.
So
Super cool. That's very interesting.
Yep, certainly more to come on the plant anesthesia side of things.
yes, yes. Well,
Yeah.
that has been a really fascinating and very interesting discussion, all the way
Hmm.
from anesthesia, consciousness, science, and also psychedelics.
Yeah.
So it's been really really fun.
Yeah, I was just gonna ask at least one more question. Just personally for you, Bruce, how did you end up in the neuropharmacology field?
Well, I was always interested in the brain since about grade 8 or 9. I saw this Life magazine back in the day when the Life magazine was huge
Mm-hmm.
and full of pictures, and they had an article on the brain, and it just blew me away. All that was known at that time of the anatomy, this part of the brain does this, and that part of the brain does that. And I thought, wow, that's really neat. Um,
Yeah.
I knew from an early age I was going to be a neuroscientist, and I wasn't quite sure what. At the time I went into university, I went into a biochemistry program, thinking that understanding the chemicals of the brain would be a good place to go. And I still believe that that's true. But working in a lab in my second year of university over the summer, I learned about electrophysiology. This is where you record the electrical activity of neurons. And this is so much cooler than biochemistry because, biochemistry can take weeks
Yes.
before you know the results of your experiment. Um, Electrophysiology is instant gratification. You know at the end of the day what the experiment did, and you're looking at living tissue that's still functioning, and
Gotcha.
so I just thought that was the coolest way to study the brain. And so I immediately dropped out of biochemistry and switched to physiology, and then went on to do a master's in pharmacology because I figured, one thing that we could learn from is drugs that affect the brain because they're going to act on the brain in some physical way. And if we can determine what that is, we can link the subjective effects of drugs, like loss of consciousness, for example, to cellular and molecular systems in the brain. And so that's what got me into neuropharmacology.
Very cool. This has been really fantastic to hear, your research and your insight on these different things, but also, try to break it down and share to hopefully new listeners, and maybe somebody will be inspired to also go into this field someday too. That would be amazing.
Well, I hope so.
Yeah.
Yes. Well, thank you so much, Bruce. This was really fun. Uh,
Yeah.
we'll do it again after we have the paper published.
Okay, and peace to both of you.
Thank you, Bruce. And finally, just thank you guys for listening or watching the Terrascience podcast. Please like and subscribe and hit the notification bell so you don't miss any of our future episodes with these fantastic guests like Bruce. Thank you.